Okay, hi everybody, I am Caroline, I'm based at the University of Manchester and I am delighted to be taking the last part of this session on entrainment to talk about frequency-dependent entrainment and plasticity with a focus on mechanisms. So I just need to state first of all that although I'm delivering this talk, it was actually a collaboration between myself, Professor Charlotte Stagg from Oxford and Professor Christoph Hermann from Oldenburg. They donated many of the slides. I'm discussing lots of their work throughout the talk and I hope I've made it clear where that is the case. So in the talk I'm going to talk about entrainment and plasticity, and the stimulation type I'm going to mainly focus on for the most part is TACS, which is transcranial alternating current stimulation. So just as a very brief introduction, here on the right you can see a kind of standard TACS setup, and it works by applying an alternating current to the brain via electrodes that are attached to the scalp, and you can use different montages to target different brain regions. And so the way it works is that, just by way of comparison, this is TDCS on the top. This is where you apply a constant current, of a specific amplitude for your set duration. And that can be applied at different polarities. So anodal is excitatory and cathodal is inhibitory. TACS, on the other hand, is an oscillating current. It's applied at a specific frequency. And you can see it takes the form of a sine wave. So it's this is TACS, and that's kind of the stimulation type that I'll be focusing on for most of the talk. And it's assumed that TACS is able to entrain the neural oscillators if it's applied at certain frequencies, phases and amplitudes. And so to just demonstrate that kind of schematically, we have this diagram here on the right. You can see the bottom and think of this as the spiking activity or the LFP data, and the top is the entraining current. And you can see that the spikes coincide with the peaks of the entraining current. And so you would consider this to be a case of entrainment. And here the opposite occurs. The spikes don't coincide with either the peaks or the trough of the entraining curve. And so you would say this is a case where we do not see entrainment. And that can be summarized in this graph on the left. This is the so-called Arnold-Tong diagram. So on the x-axis we have the driving frequency of the input. On the y-axis the strength or the energy of the driver. And the resonance frequency of the oscillator is 10 Hertz in this case. And so what you can see is that for very low input energies, you can entrain the oscillator to a frequency that is close to its resonance. But if you want to move away from that frequency, then more energy is required. And that's probably quite intuitive. And so this theory was tested computationally by Christoph's group. And so they simulated a neural network of 800 excitatory neurons and 200 inhibitory neurons that were coupled. And the connectivity architecture was structured so that the connection strengths and the delays were distance dependent. So that means that neurons that were close together were connected with high probability. So this slide here is just to show you the structure of the network. So you can see that the connection strengths are very dense for short distances, and the propagation delays were short, and that's compared to when neurons were far apart in the network, they were much less likely to be connected. And so if that network was run at rest, then it had a natural frequency of around 10 Hertz. So you can see that in the frequency plot, but you can also see it in the raster plots here on the right. That's with no kind of external input. And when they stimulated it with a 10 Hertz pulse train, so you can just kind of see that in the red behind here, this is the average activity in black, then what we get is an amplification of the response at 10 Hertz and a smaller response at the first harmonic. And you can see that in the raster plots that the activity has become much more synchronized. Finally, when they're entrained with a 17.5 Hertz stimulus, you can see actually that the maximal response is at half that frequency, 9 Hertz, much closer to the resonance frequency of the system, with a very small response at the stimulation frequency. And you can see that pattern again here in the raster plot. So when they stimulated at frequencies that were kind of much greater than the resonance frequency of the system, the biggest response was actually still at the resonance frequency. And then to summarize that data, they tested different frequencies from 1 to 100 Hertz. So you can see that across the x-axis here. and the stimulation amplitude is on the y-axis. And so this is showing the response, this graph on the left is showing the response power of the stimulation frequency, and you can see those Arnold tongue patterns at 10 Hz, but also at 20, 30 and 40 Hz at the harmonics. And again, you can see that on the right, this is showing the frequency of the response over the network. And so this work is really showing us that the importance of frequency choice with regards to entrainment. So that's entrainment, and now I'll move on to plasticity. So there are numerous studies, and I'll go through a couple of those in the coming slides, that show changes to behaviour or to the network after the stimulation is removed. And in some cases we think that is due to plastic changes in network connectivity. So one hypothesis about why TACs or any frequency-dependent stimulation, be it sensory, magnetic, electric, can facilitate plasticity is that it provides a temporal predictability or a temporal structure within which neurons will regularly fire and thus increase their connection strengths via a mechanism called spike timing dependent plasticity, or STDP. So for anyone who's not familiar, the basic idea of STDP is that you have two scenarios. If you have two neurons, a presynaptic and a postsynaptic neuron, you either have the case that the presynaptic neuron fires before the postsynaptic neuron, and that will lead to LTP, long-term potentiation, which is an increase in the connection strength between those two neurons. Or you have the case where the postsynaptic neuron fires before the presynaptic neuron does, and in that case that will lead to depression of the connection strength between the two synapses. So that's LTD. So in another computational study by Christoph's group, they looked at the connection strength between pairs of neurons. So here they created a neural network model with one driving neuron, and that was an excitatory neuron, and it was connected to a hidden layer of 2,500 other neurons, which were connected via recurrent loops of different durations. So that is, these durations are all along the x-axis here. The weights between the neurons were randomly assigned at the start and updated depending on the timing of the spikes between each pair according to this rule that I've just described. And what they found was that the synapses of those circuits that have a resonance frequency similar to that of the repetitive input, they stimulated this neuron here with a 10 Hz stimulation, and this delta t is the time of this recurrent loop, so for the time that it would take for this spike to traverse to this neuron and back. So at each point, depending on the timing of these spikes, according to the LTP and LTD rules, they adjusted the strength between each of the pairs of neurons. And what they found was that the synapses of those circuits that have a resonance frequency similar to that of the resonance frequency of the driving stimulation are strengthened during the stimulation, and after stimulation those synaptic changes persisted and resulted in enhanced neural activity at the resonance frequency of those circuits. So this dashed line here is the kind of average synaptic strength, and you can see that for these circuits in this range, the connection strength was increased above that average. So this is some more evidence now that the choice of frequency is important, not just for entrainment, but also for the induction of plasticity within our neural networks. Okay, so hopefully I've persuaded you now that frequency selection is important when it comes to inducing plasticity in cortical or neural networks. But what about more complex frequency profiles? So there is lots of evidence from animal literature, particularly from the late John Lisman's lab, that learning paradigms, especially in the context of working memory, are associated with gamma activity that is amplitude modulated by the phase of the underlying theta activity. So here you can see a theta wave, and now I'm adding in the faster gamma activity that appears and is correlated with the peaks of the theta wave. And so that is theta modulated gamma. Now, gamma theta coupling is ubiquitous across species, so this is some pilot data from Charlotte's lab that shows the same coupling in human and mouse motor cortex. So in the next study, Professor Stagg and her group addressed the question as to whether we can modulate theta gamma coupling to aid motor learning. And so to do that, they applied tax over the motor cortex to modulate theta gamma activity during motor skill acquisition. So here you can see, first of all, in A, this is the montage of the tax setup, targeting the motor network. In B, you can see these are the tax stimulus paradigm. So here we have theta gamma being modulated by the theta peak. Here we have gamma being modulated by the theta trough, and there was also a sham condition where there was no tax. On the right, this is kind of a photo of somebody's setup, and they had a baseline task. It was a thumb abduction task, and tax was applied for the first 3 blocks out of 5, and then there was another measurement taken an hour later to test for and after effects. So to look at those results then, this is the sham condition, and so this measure of acceleration is then the measure of learning in this particular task, and you can see that the participants improve their performance across all of the blocks. If you add in the trough modulated gamma wave, so this is where gamma was appearing in the trough of the theta wave, you can see that there is essentially no difference between those two curves at most time points. But then if we add in where theta was modulating gamma during the peaks of that wave, you can see that there is improved performance once you get to these later time blocks. And that persisted for these measurements that were taken an hour later. So those results suggest that driving gamma activity during the peak, but not the trough of the theta oscillations, improves motor skill acquisition. Now, the final thing I want to talk about is the effect of frequency-dependent plasticity or stimulation on neurochemistry. I'll just introduce the brain's two major neurotransmitters in advance of that. So GABA is the brain's major inhibitory neurotransmitter, and it acts on at least 2 types of GABA receptor to hyperpolarize postsynaptic cells. And glutamate is the major excitatory neurotransmitter found in the brain. It acts on AMPA and NMDA receptors to elicit firing in the postsynaptic cell. And there is evidence from animal studies that at low firing frequencies, tonic GABA levels remain high and inhibit LTP. So LTP is long-term potentiation it's a mechanism of plasticity that I mentioned a few slides back from Christoph's work. However, during high stimulation frequencies, GABA actually acts to inhibit the presynaptic inhibitory cells, depressing their activity and allowing LTP to take place. And we have evidence of that data from in vitro slice work. And now I'm going to show you a study where we found some evidence in humans. So we tested this in a simple experiment in the somatosensory system. The somatosensory system is well controlled, well understood. It has exquisite topographical mapping, and it's highly plastic. And so our stimulus was a vibrating stimulus applied to 2 digits of the right hand. So I did say that I would only talk about tax. So this is a sensory stimulation, but I will include tax in this in a couple of slides and show you the effect of adding that in. We use 2 stimulation frequencies. We can just call them high and low for now. And we tested the effects of our stimulation using different imaging and behavioural modalities. So one of our outcomes was a psychophysics measurement. We performed a tactile mislocalization test, and we found that after the high frequency stimulation only, participants failed to detect which finger was being stimulated. And this is considered to be a marker of plasticity, as we had previously found using fMRI that this same high frequency stimulation caused the digit representations to shift closer together across the cortical surface due to increased lateral connectivity, which is the cause of the impaired performance on the task. So our idea is that inhibition went down, and so that facilitated plasticity in this high frequency stimulation case. The next thing we wanted to do was to see whether we could augment this behavioural effect by applying an oscillating electrical current to the scalp concurrently with the vibrating stimulus to the fingertip. So this time we kept the frequency the same, we just used this high frequency stimulus, but instead we varied the phase of the tax and the digit stimulation relative to each other. And we found that if we applied the tax to the scalp in synchrony or in phase with the stimulus at the fingertips, we enhanced the effect on behaviour. Alternatively, if we applied it out of phase or out of synchrony, anti-phase, we abolished the effect, indicating the potential for electrical stimulation to further modulate plasticity. But going back to that initial study with the fingertips, we wanted to understand a bit more about the mechanisms underlying these changes, and so we acquired magnetic resonance spectroscopy, or MRS, to measure GABA and glutamate, the brain's major inhibitory and excitatory neurotransmitters that I mentioned a few slides back. And that's because we wanted to test this hypothesis that inhibition must reduce in order to facilitate plastic change. And we found that using the high frequency stimulation, we lowered levels of inhibition in the somatosensory cortex. And this was not the case for the low frequency condition. And this work shows the critical importance of GABA disinhibition in modulating plastic change. So I'll finish with a summary. From Christoph's work, it seemed like the maximal entrainment of a network occurs near the resonance frequency of the system. And if you want to move away from that frequency, you need more input energy. From Charlotte's work, we saw that frequency is a critical stimulation for facilitating plasticity, and that even complex frequency profiles can be accounted for in our stimulation paradigms. And from my work, we showed that GABA disinhibition may play a role in frequency-dependent plasticity, and that is something for further study. So I'll finish just by saying thank you so much for listening, and I'm happy to take any questions.